This presentation has been recorded for students of pharmacy at Keele University, and if you're viewing it from some other site or institution, you are not authorised to do so. This lecture is really a continuation of something we started to cover last semester when we looked at how medicine can be personalised for individual patients by taking into account pharmacodynamic variations between patients. In other words, the differences between patients and how they respond to a particular drug. In that lecture we were considering pharmacodynamic aspects of personalised medicine. This semester we're considering pharmacokinetic aspects of personalised medicine. And I direct you to look at a couple of these references down below. Certainly this one in systems biology and medicine. A lot of what I've talked about in this presentation has been taken from that particular reference. So I showed you this slide last semester, and it's really an explanation of why we need to think about personalising medicine. One question you might ask is, well, why can't we just give all patients the same dose of a drug? Surely that should work. And this is a quote from Alan Roses, who at the time was Vice President at GlaxoSmithKline, and he made this famous comment: the vast majority of drugs, more than 90%, only work in 30 to 50% of the people, which means a lot of the time the drugs that are being dispensed don't actually do what we want them to do. And that then leads us to think that it's perhaps appropriate to personalise the use of medicines for individuals, so a greater proportion of the time the drugs actually work. And I also showed you this slide last semester as well, but I just wanted to remind you again, this is the data that supports what Alan Roses was saying, and it shows that if you look across a range of therapeutic areas, on average the drugs are only effective about 50% of the time. So this slide summarises the link between what we covered last semester and what we're covering this semester. Last semester we thought about how pharmacodynamic differences between patients might lead to a need for personalising medicine. So for example, the IC50 of a drug or the EC50 of a drug might differ between different patients, and so you might think that it might be necessary to change the dose of a drug for an individual patient because of that. This semester we're thinking how pharmacokinetic parameters might differ between patients, and again that might lead us to think that we need to treat patients in different ways because of these differences in the pharmacokinetic properties of drugs in individual patients. So it's become feasible within the past year or so to sequence an individual patient's genome for under $1,000. And that's been set as a goal now for a number of years, and it was thought that when we reach that goal it would be feasible to use that routinely in clinical practice. And it's not hit the NHS yet, but I think we can expect to see that in the not too distant future having a patient's genome sequenced will become a routine diagnostic test. So what can we learn from that? Well, the human genome is probably the best indication we've got of differences in the response of an individual patient to a particular drug. So the genome comprises around about 3 billion base pairs of sequence, and within that we know there are something like 10 million different sequence variations. So these are sequences that vary between different patients. And I just want to provide you some terminology in case you've forgotten. If it's a single nucleotide that differs between two patients, then that's called a single nucleotide polymorphism. And that's just a complicated way of saying one nucleotide is different. This often gets called a SNP, SNP for single nucleotide polymorphism. So an example of a SNP is shown down here. Here's a sequence in one patient, and you can see there's a C in the middle, and here in another patient there's a bit of sequence. Instead of a C there's a T. Obviously this is a very short piece of sequence, I'm just using it for illustrative purposes. Now we can talk about two types of SNPs. We can talk about synonymous SNPs or non-synonymous SNPs. So you recall, I hope, that the genetic code is redundant. By that I mean that there are several ways of coding for a particular amino acids. There might be 4 codons, for example, encoding for one particular amino acid. So you can imagine, in this case we've got here, where there's a C in the middle, that might change the sequence that's coded for, the amino acid sequence that's encoded, or it might not. depending on whether this alteration in this nucleotide it still encodes the same amino acid. So if the same amino acid is encoded in these two SNPs, those are called synonymous SNPs. If, on the other hand, the change in the sequence leads to a change of the encoded polypeptide, then that's called a non-synonymous SNP. So those are just two pieces of terminology I wanted to provide you with, as I expect you'll come across them at some point in the future. So the study of how a particular patient's genome sequence affects their response to a drug is called pharmacogenetics. And so you can see that the sequence a patient inherits from their parents can determine their response to a particular drug. And when we're talking about response here, we could be talking about the efficacy of the drug, in other words, whether it does what we want it to or not, or we could be talking about the adverse effects a particular drug elicits. So I'm not going to distinguish between these two in this presentation. I'm just going to refer to those both as the response to the drug, and it should be evident whether I'm talking about efficacy or the adverse effects at some point. Now, one last piece of terminology that I'd like you to be aware of is the distinction between monogenic and polygenic. So if the response is affected by or controlled by a single gene, then we'd call that a monogenic situation. But if there are multiple genes involved in determining the response, that would be then called polygenic. Okay, so I'd like to just touch on one last point before we get into the real body of the presentation, and that's to try and understand what the point of a lot of the material in this lecture is. There'll be a lot of examples presented in the next few slides of how variations in genetic sequence can affect the response to a drug. And oftentimes students come to this and think, okay, well, my role here is just to try and memorize all these examples because they're particularly important examples. Well, in some of the cases that's true, they are particularly important examples, and it's useful to know some of them. But that's not really the goal of the presentation. The goal is to try and illustrate some principles of how genetics, a patient's genetics, can influence the response to the drug. So the examples are there to show you how that can happen. In some cases it's important to know them, but it's really understanding the principle that's the most important point. So the first example I'd like to use comes from some work that you studied last semester, and that's the effects of a drug called succinylcholine on the nicotinic acetylcholine receptor. And please go back and review the set presentations on the skeletal muscle and the somatic nervous system. In particular, you need to remember that at the neuromuscular junction, the release of acetylcholine leads to the acetylcholine binding to the acetylcholine receptor, the nicotinic acetylcholine receptor in skeletal muscle, and the nicotinic acetylcholine receptor then lets in sodium that depolarizes the skeletal muscle, that causes the release of calcium from the sarcoplasmic reticulum, and that drives skeletal muscle contraction. Now, succinylcholine is a drug that's used to cause relaxation of muscle. It can be used to facilitate surgery, or it can be used to facilitate the introduction of a tube into a patient's airway, so the succinylcholine allows the musculature to relax. And the reason it does that, it acts as what we call a depolarizing blocker. Again, please go back if you've forgotten this, and this is a little bit quick for you. You covered it last semester, but in brief, the succinylcholine is not degraded by acetylcholine esterase. Acetylcholine esterase is the enzyme that degrades acetylcholine. Succinylcholine isn't a substrate for acetylcholine esterase. It's a substrate for an enzyme called butyrylcholine esterase, and that degrades the succinylcholine more slowly excuse me, than acetylcholine esterase degrades acetylcholine. So the effect of that is that the succinylcholine has a prolonged activity excuse me, at the nicotinic acetylcholine receptor, and it stimulates the nicotinic acetylcholine receptor continually. That causes the continual release of calcium from the sarcoplasmic reticulum until the sarcoplasmic reticulum is devoid of calcium, and as a consequence there's no further muscle contraction. So it's actually, if you like, an agonist that's having a too prolonged effect, and it essentially switches off the ability of the nicotinic acetylcholine receptor to trigger muscle contraction because there's no calcium left in the sarcoplasmic reticulum. So if that's too fast for you, please go back and look at what we studied last semester. So Succinylcholine is degraded instead by an enzyme called butyrylcholinesterase. And we know of one common SNP, at least in butyrylcholinesterase, that affects the patient response to that, and that's at position 209 in the mRNA sequence. And we know that in some cases there's a guanine at position 209 in the mRNA sequence, and in other cases there's an adenine at position 209 in the mRNA sequence. So if there's a guanine at position 209 in the mRNA, that encodes an aspartate at position 70 in the protein sequence. And if there's an aspartate at position 70, then that butyrylcholinesterase is able to degrade the succinylmethonium. So after the patient receives the drug, the succinylmethonium gets degraded and the patient eventually recovers from the effect of the succinylmethonium. If, however, a particular patient has this A209 SNP, that A209 leads to a glycine at position 70 in the protein sequence, and this butyrylcholinesterase is called atypical butyrylcholinesterase because it degrades the succinylmethonium more slowly. The consequence of that is the succinylmethonium has a more protracted effect than would otherwise have expected. So as a consequence, the muscle relaxation that you get from giving succinylmethonium to a patient with this glycine 70 butyrylcholinesterase continues for a longer time than you'd expect. There's prolonged muscle relaxation, and so this means that patients might be at the risk of prolonged paralysis if they've got this particular SNP. So there we go. That's quite a striking example, I think, because it's a single nucleotide variation, either a G or an A at one particular point in the mRNA sequence, and that leads us to a significant difference in the response to the drug. Okay, so the next example I'd like to talk about is a drug called isoniazid. Isoniazid is used in the treatment of tuberculosis, and whilst it's an effective drug, there are some adverse effects associated with it, and the most common of which is something called peripheral neuropathy. So peripheral neuropathy basically means damage to the peripheral nervous system, and in this case it's caused by isoniazid, and the isoniazid can cause patients to complain of numbness or tingling or even pain in their periphery. Now, isoniazid is metabolized by an enzyme called N-acetyltransferase 2, and N-acetyltransferase 2 transfers an acetyl group onto isoniazid, and that inactivates isoniazid. Now, there are multiple SNPs, or multiple different alleles of the NAT2 gene known. In fact, there's at least 27 different alleles known, and because this is rather complicated, patients are either characterized as either being fast acetylators or slow acetylators. So that depends upon whether this N-acetyl2 transferase gene has a fast catalytic rate, in which case they're fast acetylators, or if it's got a slow catalytic rate, then they're slow acetylators. So if the patient's a fast acetylator, they can very rapidly acetylate isoniazid, and the isoniazid gets inactivated. If, however, the patients have got an allele of NAT2 that leads it to be catalytically quite slow, and the patients are then slow acetylators, they metabolize the isoniazid more slowly, and because the isoniazid metabolizes more slowly, the drug can potentially build up to a higher level, and as a consequence, those patients then are at a greater risk of peripheral nerve damage, or a greater risk of peripheral neuropathy. So whether the patients are fast acetylators or slow acetylators determines the likelihood of them suffering peripheral neuropathy. Now, this isn't just important for isoniazid. There are several other drugs that are metabolized by N-acetyltransferase 2. So, for example, procainamide, which is used in treating arrhythmia. And so it's worth understanding this concept of fast acetylators and slow acetylators, and the fact that this can control the rate at which a particular drug is metabolized, and that can control the response to a particular drug. Okay, the next example I'd like to discuss is cytochrome P450, and in particular the isoform known as 2D6, because this particular isoform is known to vary substantially between different patients. Now, if you've forgotten what cytochrome P450 does, then please go back and look at the lectures that Tony gave you, but what I hope you all immediately remember is that it's an important drug involved in phase 1 metabolism. Now, we can break down patients into two groups. those that show, well, three groups really, I suppose. Those that have what you might call common levels of cytochrome 2d6 activity, those patients who have reduced cytochrome 2d6 activity, and those patients have got increased 2d6 activity. So a patient might have reduced cytochrome P450 2d6 activity, perhaps because one of the alleles that they've inherited encodes an enzyme with a reduced catalytic activity, or maybe both of the alleles encode an enzyme with reduced activity, or they could have lost one or two alleles of that encode that gene, so that would lead to less protein being produced. So what is the outcome of the reduced activity of cytochrome 2d6? Well, one obvious outcome is that if you consider a drug that's normally metabolized by cytochrome P450 2d6, then if the activity of 2d6 is reduced, then that means that the drug is likely to have a longer half-life, and if a patient is given successive doses of a drug, then there's a potential for higher plasma levels of the drug to accumulate, and so you would then anticipate that there's a possibility of seeing an increased response to the drug, and that's actually seen with a drug called nortriptyline, which is used as an antidepressant. So that's I guess the kind of obvious expectation that you'd think of. It's the first thing you'd think of if you were talking about a metabolic enzyme that's been inactivated. But what about drugs that are prodrugs that are actually activated by 2d6? So instead of being metabolized by 2d6, what about a drug that's actually activated by metabolism by 2d6, so a prodrug? And there are examples of drugs like this. Codeine, which I'm sure you're all aware of, actually gets converted to morphine, and it's only when it's converted to morphine that codeine has its analgesic properties. And tamoxifen is a drug used in the treatment of breast cancer. It's a partial agonist at something called the estrogen receptor, and that is also activated by 2d6. So you can imagine then, if patients have reduced P450 2d6 activity, then these prodrugs will get activated more slowly, and that means the response to these drugs will be diminished. And in fact, there's a study showing that the genotype of a patient can actually influence the survival of breast cancer patients who are receiving tamoxifen, presumably because the tamoxifen is activated more slowly. So far we've considered examples where 2d6 has reduced activity. What about cases when the activity of cytochrome P450 2d6 is actually increased? Well, first of all, how could that come about? Well, one possibility that's been observed is the increased number of copy numbers of the gene. So if you've got more copies of the gene, then presumably more protein is produced. And these patients then get described as ultra-rapid metabolizers. They have a very large 2d6 activity, and so any drugs that are substrates of 2d6 are very rapidly metabolized. And so it ought to be fairly easy to predict the effects of this increased 2d6 activity on nortriptyline and codeine. So nortriptyline, remember, isn't activated by 2d6, and codeine is activated by it. I'd like you to try and think about what the effect of increased 2d6 activity will be, and in particular think about why patients who have got increased 2d6 activity might show morphine-like toxicity if they're receiving codeine. Have a think about that, and we'll discuss it during the teaching session. Okay, the next drug that I'd like to cover is 6-mercaptopurine. 6-mercaptopurine is a drug that's used to treat leukemia. It's an example of cytotoxic chemotherapy. It's called mercaptopurine because it's a purine, as you might expect, and the mercapto part, mercapto generally refers to sulfur, and you can see a sulfur group here on the drug. And the enzyme that inactivates it, thiopurine S-methyltransferase, or TPMT for short, the thio part refers to the sulfur as well. So it's a methyltransferase, so you can see the methyl group here that's been added to the mercaptopurine. So that's just trying to get that bit's not particularly important. I just wanted to try and understand some of the terminology there. The bit that is important is that mercaptopurine is used as cytotoxic chemotherapy to treat leukemia. The way it works is it blocks the synthesis of purine nucleotides, and if you think about cancer cells, they generally proliferate in an uncontrolled fashion, and in order to proliferate they need to go through the cell cycle. Then going through the cell cycle, they've got to replicate DNA if they're going to replicate their DNA, they need to have nucleotides to synthesize the DNA, and the way mercaptopurine works is it blocks the synthesis of purines, so there's not enough purines available for the cells to synthesize DNA, and so they arrest as they're going through the cell cycle, and then normally they undergo cell death. So it works by blocking the synthesis of purine nucleotides, so the cells can't replicate. So because it's cytotoxic chemotherapy, like many other chemotherapeutic agents, it's got a very narrow therapeutic window, and the major adverse event associated with 6-mercaptopurine is myelosuppression. So what does myelosuppression mean? Well, if you remember studies that you've done of the immune system, there's the myeloid lineage and the lymphoid lineage. The lymphoid lineage refers to cells that produce antibodies. The myeloid cells are cells such as neutrophils and macrophages, and the reason that the mercaptopurine causes myelosuppression is it inhibits the precursor cells that differentiate into myeloid cells, inhibits their replication, because in order for them to replicate, they need to replicate their DNA as well. So these precursor cells are unable to form myeloid cells, and so the precursor cells are unable to replicate and form the myeloid cells, and so we see less myeloid cells being produced. So patients will typically have a reduced neutrophil count. So you can imagine then that if a patient has a variation in their TPMT activity, that change, the autometabolism of the mercaptopurine is going to affect the likelihood of myelosuppression being observed in a patient. So this shows the organization of the gene encoding TPMT, and so the bits shown in green are the exons, and the bits shown in white are the introns in the TPMT gene. So normally the exons will be spliced together to produce the mRNA, and that mRNA is then translated into the protein. Now we're aware of a number of SNPs in the TPMT gene. The first one is seen at position 460 in the mRNA, and there can either be a G there or an A. So normally it's a G, but sometimes an A is observed, and if there is a change from a G to an A at position 460 in the mRNA, that leads to a change in the protein, changing from an alanine to a threonine at the 154th amino acid in the protein sequence. There's another SNP that we're aware of, that's at position 719 in the mRNA sequence. Normally there's an A there, but sometimes a G can be observed, and if that occurs, that leads to a change in the protein sequence, and instead of a tyrosine at position 240 in the protein sequence, we now have a cysteine. Both of these changes, the alanine to the threonine and the tyrosine to the cysteine, reduce the catalytic activity of TPMT, and so you can expect then that if any of these changes occur, the reduced catalytic activity of TPMT is going to increase the likelihood of those patients suffering myelosuppression. Now, how often do we see these SNPs? Well, in some patients we see just the 719 SNP. So very often in Asians, but rarely in Caucasians, we see the A719 to G SNP, but not the G460 to A SNP, and we call that allele the TPMT3C allele. So in the TPMT3C allele, you've got G at 460, but you've also got G at 719. There's another allele called the TPMT3B allele, and that's kind of the opposite to the previous one. So in those patients, you see a G460 changing to an A, so there's an A at position 460 in the mRNA sequence, but the A is retained at position 719. And then lastly, sometimes you see changes at both of these sites. So you see a change from a G to A at position 460, and a change from an A to a G at position 719. And that particular allele, where both of these SNPs are observed, is observed in Caucasians, but rarely in Asians. So it's interesting then to note that not only do we see multiple changes in this particular gene, but those changes seem to be reflected in the ethnic background of patients. So this is a monogenic situation because at present we're only considering one particular gene. We're just considering the TPMT gene. And why is that important? Well, if we look at how TPMT activity varies across different patients, we see what we might call a trimodal distribution. What do I mean by that? Well, to understand that, you have to remember that we inherit two copies of most genes. So we've got two alleles of most genes, and each allele could either be functional or non-functional. And so if both alleles are functional, then the patients will have high TPMT activity. Alternatively, in some patients, one allele may have been inactivated. So one allele was functioning normally, and the other copy of the gene encodes a non-functional TPMT enzyme. So in that case, we see intermediate TPMT activity. Alternatively, some patients may have an inactivation of both copies of the gene, so both alleles are dysfunctional, and then we see very little TPMT activity at all. You still see a little bit because many of the mutations that inactivate the TPMT gene don't fully inactivate it, it's only partially inactivated. It's reduced catalytic activity rather than completely blocked in many cases. Okay, so this type of distribution is sort of reminiscent of what you might call classical Mendelian genetics. So if you think about individuals with a particular gene, you might classify them as wild type, having two copies of a gene, or they might have one copy inactivated, or two copies might be inactivated, and you very often see that in classical Mendelian genetics. And the reason you see that is because there's only one gene involved. If you've got either two wild type copies of that gene, one wild type copy of the gene, or no wild type copies of the gene. There's only three possibilities there because there's only one gene involved. If there are more than one gene involved, it's a polygenic situation, then you can see that you wouldn't have these nice three simple groups. So the key point to take from this is you see this trimodal distribution simply because there are two copies of this gene, and we're only talking about a single gene in this case. Okay, the next example I want to look at is warfarin, and this provides an example of polygenic pharmacogenetics. In other words, there's more than one gene involved this time. And in this particular case, there are both pharmacokinetic and pharmacodynamic factors involved in determining the response to warfarin, so it's a little bit more complicated. So first of all, you need to go back and look at the work you did previously on warfarin and remember its mechanism of action. You need to go and recall that it's used as an anticoagulant. You need to recall its mechanism of action in preventing the synthesis of vitamin K and how that regulates clotting factors. And you need to go back and look at how it's necessary to monitor the activity of warfarin in the clinic by measuring something called the international normalized ratio to ensure patients aren't overdosed with warfarin and then at risk of excessive bleeding. You need to really go back and check that again, it's important stuff. So warfarin is metabolized by cytochrome P450. In this case, the isoform responsible is one called 2C9. And there are two variants of 2C9 which substantially reduce 2C9's activity, either a change from an arginine at position 144 to a cysteine, or a change of an isoleucine at position 358 to a leucine. And in both those cases, you can see reduced activity of the 2C9 enzyme. So you should think about what kind of distribution you'd expect to see in this particular case, remembering that there are two alleles of 2C9. So those are the pharmacokinetic factors that affect the response to warfarin, and you should ask yourself, well, what's going to happen if a patient has the reduced 2C9 activity? What is the patient likely to be at risk of, and why and how can we control that risk? Go back and think about the INR monitoring when you're thinking about that. So those are the pharmacokinetic factors. What about the pharmacodynamic factors? Well, if you recall, warfarin inhibits the synthesis of vitamin K. It inhibits an enzyme called vitamin K epoxide reductase, and some alleles of VKORC are very sensitive to warfarin and others are less sensitive to warfarin. And so the nice simple distribution you might have predicted up here gets clouded somewhat, because when we start looking at the response to warfarin, there are pharmacodynamic factors that affect the response as well as pharmacokinetic factors. So it's polygenic pharmacogenetics because there are multiple genes that show variations that determine the patient's response to warfarin. And perhaps that's one very good reason for looking at INR, because it captures all of that simply by looking at some functional outcome by measuring the clotting time. Okay, the next class of molecule I'd like to talk about have in common the fact that they all contain a platinum a platinum atom. And you can see that reflected in the name: cisplatin, carboplatin, and oxaliplatin. The platin refers to the presence of a platinum molecule in these drugs. Now, these drugs are used as cytotoxic chemotherapy, and the way they work is the platinum in these drugs can physically attach to the DNA in the cancer cells. In other words, we can platinate the DNA in the cancer cells. When the DNA becomes platinated, platinum's attached, that prevents the cells from replicating the DNA, and as a consequence, the cells can no longer go through the cell cycle because they're unable to replicate the DNA. This leads to the cells arresting as they're going through the cell cycle, and ultimately they undergo apoptosis. So to understand the pharmacogenetics of these platinum-containing compounds, we need to understand how platinum-containing compounds are cleared, and they're cleared after conjugation to a tripeptide called glutathione. So glutathione is a tripeptide because it contains 3 amino acids: a glutamate, a cysteine, and a glycine. The cysteine, of course, contains a free sulfhydryl group, an SH group, and it's that SH group that physically can attach to the platinum in the platinum-containing compounds. And when the glutathione is physically attached to the platinum, that glutathione conjugate is then eliminated from the body by what are called ABC transporters. So these are pumps that pump the glutathione conjugate out of the body via renal excretion. So we need to look at this a little bit more depth. How is glutathione synthesized? Well, because it's a tripeptide, it's synthesized from 3 amino acids, the 3 constituent amino acids: glutamate, cysteine, and glycine. And there are 2 enzymes that regulate that synthesis: a glutamyl cysteine synthase, and that attaches the glutamate to the cysteine, and then there's a glutathione synthase, and that adds the last glycine onto the end of the glutamate cysteine dipeptide. Now, glutathione, because it's got this SH group in it, can also become oxidized. It can form a disulfide bond with itself, or rather with another glutathione molecule. So you get a dimer, if you like, of glutathione, the 2 monomers being linked by a disulfide bond through the SH group of each of the glutathione molecules. And so you might represent that as GSSG, so the 2 glutathiones linked by a disulfide linkage between the 2 glutathione molecules. And that's called oxidized glutathione. That reaction is catalyzed by an enzyme called glutathione peroxidase, and the reverse reaction is catalyzed by an enzyme called glutathione reductase. Why is that important? Well, because the oxidized glutathione is already bonded, if you like. It's bonded to another glutathione molecule, so it's not able to react with the platinum. It's only the reduced glutathione that's free to react with the platinum. So as a consequence, this oxidized glutathione doesn't contribute to clearing the platinum. So the key point is, look how many genes are involved in clearing platinum. There's 2 enzymes involved in the synthesis of glutathione. There's 2 enzymes involved in the oxidation and reduction of glutathione. An enzyme I haven't talked about yet called glutathione S-transferase is the enzyme that actually catalyzes the addition of the glutathione to the platinum, and then the platinum conjugate is cleared by these ABC transporters. So there are numerous enzymes controlling the elimination of these platinum-containing compounds. And so you can see there's a potential for this being very complicated indeed. Patients may differ in any of these genes, and those differences may have an impact on the clearance of the platinum-containing compounds, and as a consequence may have an impact on the patient's response to those platinum-containing compounds. And if you thought that was complicated enough, now let's just look at GST itself. So GST, glutathione S-transferase, that's the enzyme that's involved in adding or conjugating the glutathione to the platinum itself. There are multiple isoforms of that GST. There are over 8 isoforms of glutathione S-transferase. And don't forget, each of those, there are 2 alleles. And within each of those alleles, there may be several different SNPs. So the point is this: it's really complicated. There are numerous genes. Some of those genes have multiple isoforms. Each of the genes has 2 alleles, and in many of those genes there's numerous SNPs present as well. So it's really complicated to try and predict what the response of an individual patient might be to a particular drug. So that poses a problem for us. We want to personalize medicine, but how can we do it? Because it's so complicated. Do we have to understand the entire pathway to make these predictions? Do we have to have a full description of every enzyme that regulates the response to a drug? If so, it's going to take us an awful long time to get there. Or is there some type of shortcut we can take to try and understand what determines the response to the drug? And that's what we're going to look at next. Okay, so what can we do then to predict the response of a drug? Sorry, to predict the response of a patient to a drug. One thing we could do is identify all the genes that contribute to the response to a drug and build a complicated model, not dissimilar to the one I showed you on the last slide, populate it with mathematical data and numerically predict the response to a drug. It's feasible, but it would take a long time and it would be very challenging to do anyway, so it's probably not the best way to go. Fortunately, there's an alternative, and we call those genome-wide association studies. Let's see how those work. So we start off by running a clinical trial, and so the subjects or the patients in the clinical trial receive the drug, and then we monitor whether they respond to the drug or they don't respond to the drug. And don't forget, here we could be talking about a response as either being a therapeutic effect or an adverse effect. So we split the patients into 2 groups: those who responded to the drugs and those who didn't respond to the drug. The next thing we do is compare the genome of those sets of patients and ask, are there any features that are common to one of these groups? So for example, is there a SNP that's associated with a particular response or the lack of a particular response? If we do that, we should be able to identify genes that seem to be linked to the response to a particular drug. This is a very pragmatic approach because we're not trying to identify all the genes up front and then predict what's going to happen. Rather, what we're doing is starting off, seeing what happens, and then looking to see which genes correlate with the response. Of course, I spent quite some time with you in this last semester talking about what correlations really mean and how misleading they can be. So what we really need to do is do something to convince ourselves that correlation is real. So suppose now we've identified a particular SNP that's associated with a response to a particular drug. Well, now we need to confirm that's true. So what we do is take a second cohort of patients and divide those into 2 groups. This time, those who've got the particular SNP and those who haven't got the particular SNP. And then once we've made that prediction up front who's going to respond and who isn't going to respond, we run a second trial giving the drug to the patients and seeing if our prediction is correct. So that's really testing whether our prediction is correct. So in the first phase we were just looking for a correlation. In the second phase we're trying to predict what happens, and it's only when you can predict what happens that you can have any real confidence that your associations actually worked. So what we're trying to do then, if it's a little bit confusing, the take-home message here is we're trying to make an association between the occurrence of a particular SNP and a particular clinical response to a drug. Okay, so this is an example of a genome-wide association study, and it's looking at a class of drugs called statins. Statins are used to inhibit the synthesis of cholesterol. They're used in patients who have got elevated plasma cholesterol, and that can be a problem because the buildup of cholesterol can lead to atherosclerosis, and that can lead to arterial disease, which can potentially lead to heart attacks in the worst case. So statins are often used to reduce plasma cholesterol by inhibiting the synthesis of cholesterol. But one of the problems with statins is they cause something called myopathy. You might want to think of that as muscle pathology. In other words, the patients report pain in their muscles. They may have stiffness, they may have significant pain. In fact, so much so it might cause the patients to stop using the statin. So it's potentially a significant concern. So to look at that, a genome-wide association study was done looking at 6,000 patients, and of those patients, 85 were found who had got myopathy. And it turned out that many of those patients with myopathy had a particular SNP in a gene called OATP1B1. I'll tell you what OATP1B1 does in a minute. It's just a For the moment, all you need to know is it's a transporter protein. But anyway, it was found that a SNP in this particular gene seemed to be associated with myopathy. In other words, more patients who had myopathy had a particular SNP in this particular gene. And so the next thing to do was to run a confirmation study to confirm this prediction, and the results of that are shown in this particular study over here. So don't forget there's two copies of most genes, so there's two alleles. So at each allele you could have a different base pair. So the SNP that was identified was the 521 T521C SNP. So thymidine at 521, or a cytosine at 521 in the mRNA sequence. And what was found, if you looked at the patients, that if at one allele you had a T and at the other allele the patient had a T, then the patients were unlikely to have myopathy. If at one allele they had a cytosine and the other allele they had a thymidine, then there was intermittent likelihood of getting myopathy. Whereas if they had a cytosine at both alleles, in other words, both copies of the OAT-P1B1 gene had a cytosine at precision 521, in that case they were more likely to get the myopathy. So this then really confirmed the prediction and suggested that if patients had this T521C SNP, they were at higher risk of myopathy. So why does that come about? Why does the presence of this T521C SNP seem to cause, or seem to correlate with myopathy? And this is a model to explain that. So this is the small intestine. Statins are used, are normally delivered orally. So the statin gets absorbed from the small intestine, travels through the portal vein and into the liver. The liver is actually the major site of cholesterol synthesis. And so the drug actually gets taken up into the liver by this OAT-P1B1 transporter, and that's where the statin inhibits cholesterol synthesis. So that's a good thing from a therapeutic perspective. So the OAT-P1B1 gene in which the SNP was identified is a transporter. It's the transporter, or one of the transporters, involved in removing the statin from the blood and taking it up into the liver. However, it turns out that if there's a C at position 521 in the mRNA, then that encodes a transporter that's a less good transporter, if you like. So it's less effective at pumping statin from the blood into the liver. And so that means that there's more statin available in the blood, because statins undergo a large amount of first-pass metabolism. So if the drug isn't pumped into the liver, then the drug undergoes less first-pass metabolism and more of it makes it to the systemic circulation. Well, if more drug makes it to the systemic circulation, that means more drug can get to the muscle tissue. And so this 521C SNP is really causing the myopathy by preventing the drug being pumped into the liver so it doesn't get metabolized, and instead the drug makes it into the systemic circulation, gets to the muscle, and there causes the muscle damage. And that we think is the explanation for why patients who have got this 521C SNP are more likely to be at risk of myopathy. Okay, the point of this slide is not that you go and learn all the examples. By all means do so if you want to do so. It'll be fun, I'm sure. The point is just to give you an impression that now this is really becoming reality. We are making these correlations between genotype and pharmacodynamic or the pharmacological response. And there are several examples of drugs where we can relate the response to a drug to the genetic background of the patient. And I think we're just going to see more and more and more of this as sequencing of the human genome becomes a more frequently used diagnostic tool. So the last example I'd like to show you is a database. It's called the Pharmacogenomics Knowledge Base, and there's a link to it here at the top of the screen. Please just go and explore this database. Click on the link or copy the link and go and look around there. There's an awful lot to see. You don't have to go and learn anything or memorize anything. Just go take a look and see what's there. It lists numerous drugs. It says 647 on this slide. I think the last time I looked there were 700 something drugs that have got annotations where we can see the impact of pharmacogenomics on the response to the drugs. It's an invaluable resource. Please just go take a look at it. There's a bunch of other things there you might find useful. For example, there are biological pathways drawn there. It's an absolutely wonderful resource. Just go take a look and look around. So the last point I really want to make is what I've been trying to say here. Of course, we're going to set exams and ask you questions and so on. So oftentimes students say, do I have to learn all the examples? Why do I have to learn all these examples? Are they particularly important to know? As I said at the beginning, it's the principles that are a key thing here. But we're going to set exams, and we set exams and we're going to ask you questions. So, you know, these are potential exam questions in this lecture. So you have to learn it in order to understand the principles, I suppose, and get through exams. But the key thing you need to get more than anything else is understand the principles and the points we're trying to make. So what I'd suggest you do is go through it and try and make some generalizations for yourself. So, for example, polymorphisms can either increase or decrease metabolism, and think about ways in which they could do that. So we talked about some examples with P450, for example. Just go through the slide and make yourself some generalizations, and then think of some examples that we've covered that represent those generalizations, and then with a little bit of luck you'll find you've learnt it all without too much effort.